This methodology uses the integration of different indicators to assess the degree of transformation of any cell model. This technique provides researchers with a set of time-consuming but simple-to-use tools for assessing cell transformation in vitro. This allows the method to be used in most laboratories. Demonstrating the procedure will be Teresa Anglada, a postdoctoral researcher from my laboratory, and Joan Repullés, a microscopy specialist.
When the breast primary epithelial cell culture reaches 90% confluency, transfer the supernatant from the culture flask to a 15 milliliter tube containing 2 milliliters of fetal bovine serum and wash the cells with PBS. Treat the cells with 1 milliliter of trypsin for 5 minutes at 37 degrees Celsius. When the cells have detached, inactivate the trypsin with the reserved medium and collect the cells by centrifugation. Flick the tube to resuspend the pellet and add 1 to 2 milliliters of fresh medium for counting. After determining the cell concentration, add 300,000 cells to a new tube and bring the final volume to 5 milliliters with additional fresh culture medium. Seed cells into a new T25 flask and return the cells to the cell culture incubator. Then use the formula to calculate the number of accumulated population doublings. To assess the